right, so before starting, I just want to acknowledge some of the people behind this work, starting first with my postdoc advisor, Molly Shigorsky. And then as I hope will not become immediately obvious, I'm not a coral biologist, and so our collaborators, Nina and Nisha, serve as our general introduction into this system and continue to play critical roles over the course of this work. So coral reefs are vital ecosystems. Despite covering less than 1% of the ocean floor, they harbor more than 25% of all marine biodiversity. However, recently, reef conditions worldwide have been rapidly declining. Reefs themselves are primarily made up of a diverse group of marine invertebrates called stony corals. An example here is the crop of milliflora. These are colonial organisms, so individual animals are known as polyps, and they can reproduce asexually to form a multi-polyp colony. They're vibrantly colored when healthy. What gives them their color is an association with an intracellular dinoflagellate or algae that's a symbiont from the genus Symbiodinium. And through photosynthesis, these symbionts provide up to 90% of the energy requirements of the coral host. However, when the coral becomes stressed, it can expel the symbiont in a process known as bleaching, leaving the coral this pale white color. And in periods of prolonged bleaching, it leaves the coral essentially starved and can cause its eventual death. So mass bleaching events have been drastically been impacting reefs worldwide over the last several years. An example in the Great Barrier Reef, or GBRs. This is a map showing the average mortality during 2017. And the primary culprit of recent mass bleaching outbreaks is extreme heat. So here we can see that in regions exposed to the greatest increases in temperature were also the same regions that experienced the greatest losses in coral cover. Despite being ravaged by mass bleaching events over the last several years, there's actually quite a bit of phenotypic variation in natural populations and bleaching response. So some individuals will completely bleach, while others appear much more tolerant. And variation in thermal tolerance has actually been demonstrated to have a genetic basis in the coral. So in work published by Nina and Nisha in 2015, they took individuals at two opposite ends of the GBR and made reciprocal crosses from them. And what they found is that in larvae having parents from the warmer populations, shown here in red, had a significantly increased odds of survival in thermal heat stress experiments in the lab. Importantly, this work was conducted in larvae, which have not yet acquired their symbiont, so this effect was due to the coral genetics. And so we became interested in this, knowing now that there's strong evidence that there's a genetic basis for variation in thermal tolerance, and therefore likely bleaching, to see if we can import genome-wide association studies or GWAS-based methods developed in human genetics into this coral system to develop a genomic predictor, or so-called polygenic score of bleaching response. And so how do we go about setting up such an approach? Well, so we need a large number of phenotyped individuals. We need a high-quality reference genome. And then for a cost-effective whole genome sequencing strategy, we could sequence a subset of these collected individuals at high coverage and the vast majority at low coverage, and use those high coverage individuals to impute or predict missing genotypes in their low coverage samples. And so once we have genotypes across the genome for this large collection, we can perform an association study and estimate things like SNP heritability and construct our polygenic score, which in the simplest form just sums across the effect sizes of associated variants above some significance threshold. However, the major problem when we started this work is we really had to start from the very beginning. So we lacked a reference genome, we didn't have large collections, and we lacked just some basic population genetics parameters of things to even know if our imputation or sequencing approach would be successful. So for the rest of the talk, I'm going to go through some work over the last year or so and hopefully getting rid of this large X that existed over our approach. So our first challenge was to build a reference genome. So we took a hybrid assembly approach, combining both PacBio and Illumina reads. Our Illumina reads, we also used 10x chromium barcodes, which gives us some longer range linkage information. We had around 80x PacBio, 75x coverage of Illumina. Now we had to extract high molecular weight DNA from an adult, and there's this issue of the intracellular symbiont, so a proportion of the reads will be derived from Symbiodinium. So while we can't extract enough DNA from an individual larvae to assemble a genome with, we can pool larvae and take advantage of the fact that they have not yet acquired their symbiont, and use this mapping information to filter out symbiont reads from our adult DNA. And so after removing the symbiont reads, we had a de novo assembly of more than a megabase, or delivered with an N50 or median size of more than a megabase. So this put us in a pretty good spot already. We can go a step further by aligning to previously published linkage maps. So we can use this information by aligning these markers to begin to stitch together the linear order of our contigs and the chromosome scale scaffolds. So shown here is an example for chromosome 1. So we did this for the 14 millipore chromosomes. We assembled 475 megabase worth of sequence, and we have a scaffold N50 now approaching 20 megabase, and this is a chromosome scale assembly. We released this assembly for free on the Shiborsky Lab website. It's hosted there along with our detailed assembly methods. So we have a reference genome now, and so now we need to sequence individuals. And these have to be phenotyped. So it turned out that our collaborator Lena collected over 250 individuals in 2017 and recorded bleaching phenotype data as a visual score for each sample. We now also have chlorophyll content measurements as a more direct estimate of the bleaching phenotype. And so our first step, we wanted to sequence a subset of these at high coverage. So we had a target coverage of 35x. We did this in 2 different batches. However, for the second batch, due to a fortunate error by the sequencing facility, they queued up this genome for 35x, but for the size of a human genome. So we ended up with 160x coverage for 36 of these. So after applying some filters and removing some individuals that were likely sample misIDs, we called 6.6 million biallelic SNPs across 44 individuals. So we can use this to create a reference haplotype panel to again impute genotypes in individuals that we sequence at low coverage. So this was in collaboration with Peter Indelfato and his former lab tech Julie. So we created or constructed TN5 libraries to use for low coverage sequencing. So we did this for the remainder of our samples. On average, we had about 1.5x coverage. And among these individuals, we included 36 samples that we previously sequenced at high coverage so we can assess the accuracy of our imputation. So here we're using, for an algorithm for imputation, low impute, which is developed by our collaborator, Joe Pickrell. So shown here in these lines are accuracy of imputation for different probability cutoffs for hard calling genotypes. And so this green line at a probability cutoff of 0.95, we can get an accuracy of imputation of around 94%. At 1.5x coverage, so this puts us on par with what we can do in humans using reference panels of similar size. So now we have genome-wide SNPs for 226 individuals. We can begin to look at some basic things like population structure. Well, so here's just a simple PCA with individuals colored by the reef in which they were collected from, and we can see that there's essentially little to no population structure. This is also the same story if we visualize affected migration rates using a method called AIMS here, which is just modeling the genetic similarity between populations as it decays with geographic distance. The colors aren't great here, but we get high migration across all of our collected sites. And this is presumably due to the broadcast spawning mode of reproduction of Millipora. So adult corals, of course, can't move, and when they all spawn simultaneously, the effect is that they're only breeding with those individuals in their immediate vicinity. However, once fertilized, the larvae can swim hundreds to thousands of kilometers, creating high levels of migration and effectively random dispersal. And this unique life cycle also gives us a hint of what to look for when searching for signals of local adaptation. So in a scenario of high migration over a likely heterogeneous environment, where there's likely strong spatially varying selection, theoretical models predict that this should look like balancing selection. So in loci involved with local adaptation, we should see increased genetic diversity. So here we're just simply measuring genetic diversity as pi, 10 kb windows across the genome. And our largest signal falls right in this gene called Saxon in chromosome 7. And what's remarkably interesting about this is that this gene is known to act as a chaperone for 2 different heat shock proteins, HSP90 and HSP70. These are both known to be involved in the bleaching response across a range of Megareans. So here is showing HSP90 expression as it coincides with the initial loss of the symbiont in the model Megarean aptasia. So before ending, I also want to show that we've been able to gain some insight from the perspective of the symbiont using our sequencing approach as well, and develop another complementary measure of bleaching. So as I mentioned, we have visual scores for all samples as the bleaching phenotype. However, since we'll be extracting symbiont DNA along with our coral DNA and sequencing this all at once, we can use the total set of reads and map these to our millipora assembly, as well as draft symbiont genomes, and use the relative proportion that map between these as another measure of bleaching, since this should relate to the abundance of the symbiont in the coral tissue. So here we can see we get a pretty strong correlation with the visual score using these proportions of read mapping. Importantly, there's multiple species of symbiont that are known to associate with millipora, here called clade A, B, C, and D. And so we can use a similar mapping approach to estimate the relative proportions of these different symbiont types in each sample, which is represented in each of these dots. We can see that most individuals are dominated typically by a single species, and those that are most tolerant and have the highest bleaching scores tend to be those dominated by clade D. This is consistent with what's known in millipora and other acroprid species, where clade D seems to confer some degree of higher heat tolerance. And so we've recently now expanded this analysis out to all of our sequence samples, and this is work that's been led by an undergrad who's been working with me so far this summer, Jihan. And so here we have our bleaching phenotype measured as chlorophyll content, and then read abundance on the x-axis. And so again, we get a fairly strong relationship. And now we don't know what the amount of phenotypic variation that can be explained by this difference in the dominant symbiont species. And so we can actually estimate this in our GWAS model and estimate the proportion of variance in bleaching response that can be explained by these differences in symbiont type. And so in a basic regression framework of an association study, we can simply model the amount of variation that can be explained by genotype as a mixture of covariance and error. And so just in a simple regression framework, we can model our bleaching phenotype as being contributed by the coral genotype, the dominant symbiont species, and we can also include an interaction term to test for interaction effects between the coral genotype and the dominant symbiont species. Other covariates that we can include in our model are environmental variables that we have for each collection site. So we have over 40 for things like temperature, sediment quality, salinity. And so just very briefly, I want to show some very preliminary GWAS results using this basic model. And so here's a Manhattan plot showing the association with bleaching phenotype. And each variant here is represented as a dot. What was striking to me is that in this basic preliminary analysis, our strongest hit genome-wide falls right this exact same gene that I mentioned a few slides ago in Saxon. So already this looks extremely promising. We've also started now testing the predictive accuracy of our polygenic score. So here, prediction accuracy is measured as incremental R squared. In other words, how much better are we doing by adding in a polygenic score? This is on the x-axis p-value thresholds for a level of significance to consider variance. We get the largest predictive accuracy by considering sites above a significance threshold of 10 to the minus 4 or greater. So again, this is all very preliminary, but so far things look like they're working and seem fairly promising going forward. So just to wrap up, this is some of the work that we've done over the last year to get us to the point now where we can start to apply some of these GWAS methods and hopefully construct an accurate predictor of the bleaching response phenotype or a polygenic score for bleaching. So with that, thanks, and if there's any questions, I'll take some. Yeah.
There's some great coral work going on where we're taking heat-tolerant corals and trying to seed them with less heat-tolerant corals by breaking them off and putting them in decks and trying to marry them together. Do you think that's a shift in the clade from like D to C, or do you think that's more of like a basic architecture of the coral itself?
Yeah, I guess that's it's hard to say. I mean, I can see it being explained by both possibilities. Yeah, so there's certainly like trade-offs from the different symbiont species that associate with it, but.
Yes, do you know if there's an increased cost for clade D versus clade C?
Yeah, so typically they tend to have lower growth rates.